There is a growing body of evidence suggesting that probiotics, especially next-generation strains, may do more than improve digestion or reduce gut inflammation. Some of these targeted microbes appear capable of directly enhancing what we might call the neurobiotic sense, the ability of the gut to detect, interpret, and transmit signals about food, safety, and energy needs to the brain via neuropods and the vagus nerve. This emerging concept challenges the old idea that probiotics are simply passive passengers in the gut. Instead, they may function as active co-engineers of brain-relevant perception, helping to regulate appetite, mood, reward processing, and weight management from the inside out. To understand how probiotics might enhance this neurobiotic capacity, we need to examine their interaction with neuropods, specialized gut cells that form synapses with neurons and send real-time signals to the brain in milliseconds. Neuropods don't just detect nutrients, they are also responsive to microbial cues. Certain probiotic strains can influence the release of neurotransmitters like serotonin, GABA, and dopamine precursors, as well as the secretion of hormones like GLP-1, PYY, and CCK, all of which influence hunger, satiety, and emotion. For example, strains of Lactobacillus rhamnosus have been shown to modulate GABA receptor expression in the brain via vagal pathways, reducing stress-induced eating and promoting emotional regulation. Bifidobacterium longum has demonstrated anxiolytic and antidepressant-like effects in animal models, effects that vanish when the vagus nerve is severed, confirming the critical role of gut-brain communication. These microbes may be acting, in part, by enhancing or stabilizing neuropod sensitivity, improving the fidelity of gut-derived neural signals that the brain uses to guide behavior. The metabolic outputs of probiotics are another important factor. Many strains produce short-chain fatty acids, SCFAs, such as butyrate, which not only fuel colonocytes and improve barrier function but also modulate the excitability of vagal afferents and enteroendocrine cells. Butyrate in particular has been shown to enhance satiety signaling by increasing GLP-1 and PYY production, potentially improving post-meal satisfaction and reducing the drive to overeat. Some probiotic strains, including Faecalibacterium prosnitzii and Akkermansia mucinifila, support this process indirectly by promoting the growth of beneficial bacteria that optimize the gut's sensory interface. Where things get especially promising is in the field of precision psychobiotics, engineered or carefully selected strains aimed at specific neural or behavioral outcomes. These may include Clostridium butyricum for butyrate enhancement, Bacteroides uniformis for fat metabolism regulation, or even designer E. coli strains programmed to secrete appetite-regulating peptides. These organisms don't just support general gut health, they may directly tune the neural circuits that underlie cravings, reward sensitivity, and even emotional eating. Importantly, enhancing the neurobiotic sense isn't just about increasing satiety. It's about restoring a trustworthy internal signal environment, one in which hunger and fullness feel clear and accurate, where emotional urges are less likely to hijack appetite, and where the gut-brain dialogue supports long-term metabolic balance. In people struggling with obesity, for instance, this signaling may be chronically disrupted by inflammation, gut barrier damage, or dysbiosis. Targeted probiotics may help recalibrate these pathways, improving not only metabolic parameters, but also the felt experience of appetite regulation. Clinical research is starting to reflect this potential. In human trials, multi-strain probiotics including Lactobacillus gasseri and Bifidobacterium breve have been associated with modest but consistent reductions in body fat and waist circumference. Some studies also report improvements in leptin sensitivity, reductions in ghrelin, the hunger hormone, and lower subjective appetite ratings following probiotic use. These effects are modest on their own, but become more meaningful when integrated with dietary fiber and prebiotic intake, which can amplify microbial viability and metabolite production. Still, we are only scratching the surface. One of the biggest challenges is that probiotic efficacy is highly individualized, shaped by baseline microbiome composition, host genetics, immune status, and diet. What enhances the neurobiotic sense in one person might do little in another unless the surrounding microbial community supports integration. This is why next-gen therapies increasingly aim for symbiotic strategies, combining targeted probiotics with specific fibers like inulin, arabinoxolin, or resistant starches that selectively feed those strains and stabilize their function in the gut ecosystem. In the future, we may see a new class of microbial therapeutics that go beyond the probiotic shelf to act as neuromodulators, restoring the clarity of gut-derived cues for those living with obesity, disordered eating, or stress-related food behaviors. These interventions may be tuned to amplify neuropod responsiveness, support vagal tone, and optimize the gut's output of behaviorally relevant signals, without the need for external appetite suppressants or restrictive diets. In conclusion, yes, probiotics, especially next-generation strains, do hold the potential to enhance the neurobiotic sense. They may improve the quality of communication between gut and brain, rebalancing the physiological signals that regulate appetite, stress, mood, and long-term weight. The future of metabolic and emotional health may lie not only in what we eat, but in which microbes we recruit to help us interpret that food in a way our brains can understand and act upon.